Okay, I think we can get started. It's
Okey-dokey.
4:03, and I want to make sure that you have, good amount of time uh, for questions. So
Okey-doke.
I will start with more formal introductions. So it is a big pleasure, James, to have you here. And, it's our second attempt to have you speak at UCSF. It's still not the one I was hoping for, but hopefully third time will be the charm where you actually can come here in person. Dr. James Murphy is currently an associate professor at the Walter and Eliza Hall Institute of Medical Research, or WEHI for short, in Melbourne, Australia. And James also holds a position of a head of the Division of Inflammation at WEHI. He wears many hats, and some of them also are being on the editorial board of JBC and being an associate editor of Biochemical Society Transactions. So James was trained as a biochemist and structural biologist, and I think at heart he remains one, despite the fact that now ventures in all these other areas and technologies. And he's one of those special structural biologists who always sees in structures something that no one else can see at first, and turns it into beautiful explanations of molecular mechanisms. And James obtained his undergraduate degree with honors at University of Canterbury and PhD at the Australian National University. During his PhD, he worked with Dr. Yan Yang on structural basis for activation of interleukin receptors. And following his PhD work, James went for a couple of years to Toronto to work with Tony Passan on structural understanding of FF domains in transcription factors. And after that, came to WEHI, where he has been ever since. Initially, James held a position of senior research officer at WEHI and worked with a lot of amazing WEHI faculty like Jeff Babon, Peter Zabotar, John Silk, Isa Losset, and made a lot of contributions in a variety of fields, contributed to studies on JAK signaling and SOX signaling. But his landmark paper was published in 2013, where he identified a new critical factor of necroptosis response to be this protein called MLKL pseudokinase. And soon after, James became a laboratory head at WEHI and really single-handedly moved forward the field of understanding how MLKL is structurally regulated, how it's activated, and how its signaling intersects with the system of other kinases important in necroptosis, RIPK kinases, and I'm sure he's going to tell us a little bit about that today. He has won a number of prizes for his achievements, including Erskine Fellowship and a Burnett Prize as a top science prize at WEHI. But I want to say a few things about James and pseudokinases, because it's not an accident why James has a hashtag pseudokinase on Twitter and owns it. He organized the first pseudokinase meeting, which happened to be an international meeting, although it was very, very small, back in 2010 in London. Only really few of us had showed up who were aficionados of these unusual kinases back then. And it was a very special meeting, and James really took upon himself and took that stewardship of getting this meeting going throughout the years, which now really expanded to a large community of people who are interested in these unusual enzymes. The meetings really now expanded to pseudoenzymes and welcomed people from other fields. And James really was responsible for building this community and is still making sure to bring us together every couple of years and expanding this really exciting and ever-growing area of research. So with that, I'm going to finish to, as I promised, let you speak as well. It's really a pleasure to have you here and looking forward to your seminar.
Thanks so much, Natalia. That's a really generous introduction, and I guess I should take your lead and just plug the next meeting, which will be in 2022 in the UK, all things going to plan. So as Natalia mentioned, we really got interested in pseudokinases through work on MLKL, and I'm going to tell you about really a serendipitous story that really pushed us in this direction to begin with. And this is a long time ago, the order of 13 years ago. And I had this interest in pseudokinases, which sort of came from when I was in Toronto and working with Tony and with Frank Zakarian. Someone you know, Arvind Dar, was a PhD student in Frank's lab and doing some really interesting work with pseudokinases then. So I knew that they were going to be topical, and I just thought it was fascinating that there's something that we don't know what they do, but they have to do something because they're everywhere. So anyway, the one I'm interested in is MLKL, which is not only a dead enzyme, which leads to these sort of macabre pictures that I like to use and all these zombie references, but also it's involved in the cell death pathway called necroptosis, and hence the title, The Dance of Death. And it's been pointed out to me that I'm sort of like an annex to Halloween here, but also not to get too political, but this could equally be a picture of the last 4 years in the White House. Anyway, so we're interested in pseudokinases, and I'm sure you've heard many stories from your institute beyond about these things. So what are they? Well, they look like a protein kinase domain. They have this bilobal structure, but they're defined by the absence of what I like to call the Hanks motifs. So one or more of them, which is known to compromise their catalytic activity. And so there are very few examples of pseudokinases that are catalytically active. And for the most part, it seems that this residual activity is not important for their function. And this leads us to think of them, and this is sort of the Tony Pawson sort of idea of these protein interaction modules, which is something I've had a long-standing interest in. And it really begs the question that if pseudokinases do this, is this something that conventional protein kinases may also do? And to take a leaf out of our colleague Gerard Manning's book, he referred to them as the black sheep of the kinome once, and this really stuck with me. So they're quite prevalent. There's about 60 in mouse and human genomes, so about 10% of those kinomes, and they're even more abundant in the genomes of things like Toxoplasma, which has about a third of their protein kinases, predicted pseudokinases. And I was a bit gutted when it dawned on me way too late that there's a lot of allusions to zombie films, and I'd forgotten about this classic New Zealand film, Black Sheep. So being about zombie sheep, it seemed to really fit the bill. But we've really taken this mantle and probably taken it too far, to be absolutely honest, with some of our titles for reviews that we've written on the subject. A lot of work I've written with Pat Ayres, who's another person who's heavily involved in organising these meetings. I'm particularly proud of this title because it came out of the Liverpool meeting. And so not only did we have Sir Paul Nurse there, we also had an allusion to Sir Paul McCartney. But recently we'd sort of drawn all this together in this paper that came out of one of the meetings that we were organised, the one in Sardinia a couple of years ago. And this has a rather bland title, but regardless, it seems to have had a lot more traction. So maybe there's a lesson in that. But right here, front and centre, is MLKL. So this is an image from Jerome Klaus, who's an animator who runs Phospho Animation, which is a really cool company who do a lot of animations and great journal covers. So through writing these sorts of reviews, we'd sort of distilled down some basic principles of what pseudoenzymes and what pseudokinases might do. And I think the most well characterized is that they can act as an allosteric regulator of an active enzyme. And this is quite frequently observed. The one that we've worked on in this regard is JAK2, but you can imagine that there's a duplication event and some like protein then adapts a function that's non-catalytic, but as an allosteric regulator. So other examples from Natalia's work are things like HER3 interacting with the active HER2 to regulate its function. Other common functions include acting as scaffolds to assemble signaling complexes. So some of the work we did with Peter Mace, looking at TRIB1, where it brings together an E3 ligase and a substrate to allow modification of the substrate, but also some work with Isoleucine and Anisha Patel looking at SGK223, and this also applies to PEAK3, which is the work that Natalia has been doing, where the pseudokinase or pseudoenzyme can act to bind to another protein to regulate its localization in cells. The protein we've been principally interested in is MLKL, and we like to think of it as one of these rare beasts, which is a molecular switch or a signal integrator. So this idea that it can be modified in some way which changes its function, and that's something I'm going to tell you a bit more about coming up. When we first got into this area, which is about 13 years ago, I'd tell people with some excitement about what we were doing, and they'd say, oh, they're just acting as competitors or substrate traps. And then when we come to write up these reviews, it turns out that these functions are very infrequently observed in nature. So we were particularly excited to report last year a viral form of MLKL that we worked with Jared Manning to characterize that acts as a competitor in cells. So this is kind of the unicorn and probably arguably the least interesting, except for its scarcity. So as I mentioned, we're principally interested in MLKL. And we've been studying this for some years now. So it's from Natalia's review, but on Jared's kinome tree, it sits here, and despite its position, is probably most closely related to RIP1 and RIP3, who are two bona fide kinases, as well as two pseudokinases, ARAC2 and ARAC3. And so you can sort of tell by the name it was given, mixed lineage kinase domain-like, that when it was annotated, no one knew what it did. And that's true until 2012. We were trying to determine its function. And I'm going to give you a serendipitous historical story of why we were interested in its function. And it was only in 2012 when it was implicated in this new cell death pathway, necroptosis. And going into the study, what we knew was that it was going to be catalytically inactive because it lacked two key catalytic motifs. And it had this peculiar N-terminal domain that seemed unique. But it was these two stories that set us on a path. One from Xiaodong Wang's lab and another from subsequently from Zhenggang Liu, which implicated our protein of interest in this very cool and topical cell death pathway, program necrosis or necroptosis. And just by way of description, I'm sure you're very familiar with apoptosis, which is a caspase-mediated cell death process where cells shrink and the membranes bleb, and then they're basically swallowed up by patrolling cells such that they don't cause any deleterious effects to the organism. So this is a very important developmental process. I'm sure you've seen this described at length, and it's also a core interest of our institute. But for instance, you would end up with webbed fingers if apoptosis didn't work in your development. But necroptosis, on the other hand, is a caspase-independent process, and it's designed to perform a very different function, which is by the cells swelling, they eventually explode and release their contents, which acts to elicit an immune response. So it serves a completely different function. It's not benign to the immune system. And this has really led to this concept that it's involved in it's arisen as an ancestral host defense pathway. And this seems to have occurred also convergently in plants. Recently, there was a report from Takeki Mikawa from Cologne describing a plant MLKL family that's involved in host defense in plants. And there are a number of pathogen proteins that are known to target components of the necroptosis pathway. So it seems to fit that it would have arisen as an ancestral host defense pathway. However, our interest principally these days is that when the pathway dysfunctions, this leads to or contributes to human disease. And this has been widely implicated in a number of studies over the years, principally in animal models. We're at the moment particularly interested. There's a cool study that we're working on with a collaborator in Qatar, where a patient variation contributes to diabetes. There have been a number of studies. I'm going to tell you a little bit more about the work of Joe Hildebrand and Maria Kaupi coming up. But we're also interested with colleagues in Kiel. We've been interested in acute kidney injury, and particularly the role of if you can imagine, this cell death occurs in the case of organ transplantation, such as in kidney transplants, but also widely implicated in things like infarct pathologies. So this is really up to the minute sort of understanding, or at least our thinking of the pathway. So the pathway can be triggered by ligation of death receptors. So in the lab, we typically study the TNF receptor, but you could also, we sometimes study things like toll-like receptor 3 and 4, which are pathogen detectors, and others have implicated the intracellular pathogen detector, ZBP1 or DAI, as a receptor that promotes cell death. And so in scenarios where this family of E3 ligases, the IAPs, are disarmed, and when the proteolytic apoptosis enzyme caspase-8 is compromised, we can see assembly of this complex here, the RIP1, RIP3 complex. There are a number of other components that seem to vary depending on the cellular context, but these are the core components of what's called the necrosome, which is a cellular compartment. Perhaps this is something that we're interested in knowing more about. It may be a liquid-liquid phase separation sort of organelle, but this is, of course, highly speculative. So following initiation of the pathway, MLKL, which resides in a dormant state, we believe, in the cytoplasm, is recruited to the necrosome, where RIP3 can phosphorylate the protein and trigger what we think is a structural interconversion that allows its dissociation from RIP3. And this assembly of multiple MLKL molecules is then actively trafficked to the plasma membrane, where it forms into these what we call hotspots. And it's this, when this threshold is succeeded, that we see membrane disruption, and this causes the swelling and lysis of cells and the release of the contents, which elicits an immune reaction. So what I'm going to go through today is really a summary of our reasoning for why we think these 3 checkpoints occur. So this is very contemporary thinking. Some of it's recently been published, some of it is now in revision as of about an hour ago when we got reviews. So I just wanted to give you an historical overview. So really our interest in MLKL started a long time ago on the basis of an ENU screen. Subsequently, we tried to rationalize our findings using structures, and this is something that we've persisted with doing. And, you know, armed with a lot of recombinant protein, this gave us a platform for generating some new reagents, which have allowed us to perform more detailed imaging over the past couple of years. So I'm going to give you, first of all, an insight into why we were interested with this mouse study, and then focus on the more molecular stuff. So originally, this was a screen set up by Warren Alexander, Nick Kohler, Ben Kyle, Doug Hilton at our institute some years ago, looking for regulators of platelet synthesis. So this was an ENU mutagenesis screen using a mouse strain that Warren generated a long time ago, which is the thrombopoietin receptor knockout. So unlike in humans, which that would be lethal, in mice, they have about 10% of the normal amount of platelets and are quite healthy to a certain extent. So what you can do is use this as a platform for a mutagenesis screen and identify modifiers that can rectify this reduced platelet count. And so one of these alleles was the PLT15 allele, and it was mapped after a long slog by a number of people, including Ian Majewski and Melanie Barlow in the Institute, to a very highly conserved residue, which at the time we didn't really know what this did. And then it was our structure that really allowed us to identify this as being part of what we call the 2-helix brace region. And so this residue is engaged in contacts with the 4-helix bundle domain. And so I should point out that the 4-helix bundle domain is the executioner domain of MLKL. And so I'm not going to go into that in any detail. That was published some years ago now. But by disrupting this regulatory interaction, we could imagine that this D139V mutation would turbocharge MLKL to kill cells. So this is something that we explored using these mice. This is a project that has recently been published after a long gestation, if you will. But the thing that's always been confusing is that, so the homozygotes for this PLT15 mutation die within about 3 days of birth. And it's always been a mystery why that would happen. One of the things that would characterize in a very detailed way is the hematopoietic stem cell function. And we know that they're defective in their ability to reconstitute irradiated recipients, but also this accounts for the platelet phenotype that we saw. But the confusing thing was that this allele was less expressed. So it seems that somehow the proteins either destabilized, or that the animals, these are plots from 3 different animals of these genotypes, and 3 different embryos, I should say. And you can see that we can't detect MLKL in the homozygote. So somehow the animal's able to negatively regulate its abundance. And then we thought, okay, this is cool. We'll just make an MLKL knockout mouse and we'll recapitulate what we find over here with this, knockout effectively. And here we've got wild type and 2 knockout embryos blotted for MLKL. And you can see that these animals are perfectly healthy, and you can see that our antibody works for a start, but also that we've got abundant expression normally in different tissues. So this was made using a conventional knockout strategy by Warren Alexander. And so this was actually quite disappointing to see that these animals were perfectly healthy, but the mystery went on. Joe Hildebrand and others were able to sort of pin down that, in fact, this mutation is a gain of function. So the D139V allele is actually promoting killing. So if she expressed wild type in a bunch of different mouse fibroblast lines, where she's induced expression in the white bars using the DOC system that we use, you can see like a token amount of death. But really this mutant allele is turbocharged. It's a constitutively activated allele. And this tells us that the animals survive to birth by suppressing expression. And we're not entirely sure what happens after that, but we do know that interferons induce MLKL expression, and it could be that this is what's causing the lethality. So based on some exposure to pathogens post-birth. So Joe went on with the help of some pathologists, the Australian Phenomics Network, to characterize how the animals are dying perinatally. And what she found was massive infiltration of neutrophils and other hematopoietic cells, particularly in the mouth and head. So you can see that in these H&E and CD45 stained sections. And this really accounted for the lethality. So they look kind of normal at birth, but then they start to, they don't thrive. They're smaller and become runtier before they die. So this gives us probably the first formal evidence connecting MLKL as a, with this turbocharged mutant to an inflammatory disease. So this is a very important study in my opinion. But we still don't really know why they would have defective hematopoietic stem cells. And so that's sort of an ongoing mystery. And believe me, it's not for lack of trying that we don't understand this. So it remains a mystery. Just revisiting what I mentioned before, this is the up-to-the-minute view of the pathway as far as our interpretation is. And what I'm really going to tell you about is some more of our molecular studies to understand what's happening at each of these checkpoints. So recent evidence that we've been able to generate indicates that there is a step that involves disengagement from RIP3. And then we've got several lines of evidence pointing to a trafficking checkpoint, which involves shuttling MLKL oligomers from the cytoplasm to the plasma membrane. And then a number of lines of evidence pointing towards needing to succeed a threshold in order for cells to die. So how we've been able to get at this is with 2 real approaches. So the first one is using an imaging approach. So this is the work done principally by Andre Sampson in my lab and my collaborator Edwin Hawkins' lab. And what I've tried to highlight here is we've recently submitted a paper where we've pinned down a whole bunch of reagents that we can use to monitor each step in the pathway for both human and mouse cells. So the key thing to note is that the sequence differences between human and mouse are quite substantial. So something around 65 to 70% identity between RIP3s and between the MLKLs across species. The RIP1s are a lot closer, so we can use the same reagents for both. But we think this is a very important toolbox, and so hopefully taken up by the community. But the key thing to note here is I've tried to highlight where these antibodies came from. So a number of them are commercial, but we too have contributed by generating a number using our monoclonal facility. So this sort of comes back to having high quality recombinant protein available for immunizations. So using this approach, Andre was able to tackle two key questions. So one is, how does this trafficking occur from the cytoplasm to the plasma membrane, and what happens when MLKL accumulates at the plasma membrane? And so the backdrop to this is one of his observations, and one that we'd earlier made with Maria Tanza some years ago, that there was there's a sort of a disconnection or a delay between all the hallmarks of a cell about to undergo necroptosis and it actually dying. So we see the emergence of phosphorylated MLKL, so RIP3 modified MLKL, coming up very early post-stimulus. But if you look over here at death read out by LDH release, this only really starts to really hit its straps around 7.5 hours, whereas we're seeing phosphorylation, or I can show you here, coming up around 3 hours. So there is this temporal gap, and this was a point of interest for Andre, and so he delved very deeply into how this happens. So in part using the reagents that I mentioned. So there are two. One's a commercially available phospho MLKL reagent, where it picks up the RIP3 modified site, and the other is a reagent that we generated that detects MLKL before it's phosphorylated. And so he used an imaging approach, and he was able to identify these that post treatment with a necroptotic stimulus, which I've called TSI here, so that we use a cocktail to funnel the cell death pathway towards necroptosis. So using this cocktail and treating HT29 cells, he was able to observe these clusters that sort of form in a perinuclear sort of organization in the cytoplasm. And then using the phospho antibody, he was able to detect these quite prominent hotspots, what we called hotspots of phospho MLKL. And these start in the cytoplasm, but they form much greater magnitude on the plasma membrane. And this is something that, with the help of Lockie, who generated a macro, Andre was able to quantify. And so really the essence here is that as time goes by, so the times are color coded, we see the emergence of these cytoplasmic clusters, and there's a delay until we see these hotspots on the cell surface, and it's these hotspots of phosphorylated MLKL on the plasma membrane that really corresponds to the time when cell death occurs. And so he was able to take a closer look with some super res imaging, and so this is what we argue is the first glimpse of the necrosome, the cellular compartment I was telling you about. That's platform comprised principally of amyloid forming RIP1 and RIP3 kinases. So they form amyloids via a domain called the RIM. And so this acts as a platform for activation of MLKL. And we can see in yellow the phosphorylated MLKL emerging from the basal MLKL within these hotspots. And so this is something shown here, which we can get an idea of how these, the first clues as to how the assembly occurs on the plasma membrane. So if we start with basal MLKL, it forms these perinuclear clusters, whereas the phosphorylated MLKL is principally at the junction between the cells, but also there are some spots here, some puncta in the cytoplasm. And so the model which arose from this is really, wheat germ agglutinin showing the plasma membrane staining. So really the take home here is that what we think is happening is that MLKL assembles on these necrosomes, gets phosphorylated and forms into these small vesicle-like structures, which are then transferred to the plasma membrane, where they build up into these quite large hotspots of phosphorylated MLKL, the activated form. And this is something that Andre could, I'm going to figure out how to get my video to work. There we go. He could look with super res. So you can see that in purple, magenta is these are the basal MLKL, or the pan MLKL. And in yellow, we've got the phospho, and you can see between two cells, you can see that there's this phospho-MLKL junction. And it kind of, I like to think of it as looking like a sort of a cross-section of a lotus root. So there is an idea in the field that maybe MLKL forms pores, and if so, it seems that it's quite an, I would say, irregular structure, not like something you'd see with another lytic pathway like Gasdermin D, for instance, in pyroptosis, or maybe even with back and backs pores and mitochondrial membrane permeabilization. So Andre wanted to get down to the nuts and bolts of how this happened, and so he tried a panel of inhibitors of different processes in over a period of different time frames. And what he really pinned down was that inhibiting the Golgi microtubules and actin, or turning these into a cocktail, which we call NCB, was sufficient to block necroptosis at all these time points. So while it's well underway and while you've got phosphorylated MLKL, you can still halt the process by addition of these inhibitors. And so this is something that he looked at a lot more closely. This is using the same imaging approach, but by adding NCB at 5 hours. And you can see that we can diminish the number and magnitude of clusters, and we can diminish the number of hotspots by adding it at this time point, and more importantly, halt cell death. So this tells us that there are trafficking pathways, Golgi microtubule and actin-based trafficking pathways that are responsible for moving MLKL oligomers from the cytoplasm to the plasma membrane. And this is something that he could map along by co-staining with tubulin, and you can see vesicles along these tubulin filament, microtubule and actin filaments. So this gives us some pretty clear evidence of what's going on, but which pathway? So that's the question. So Andre did this by elimination. So by doing a lot of co-staining with a lot of different markers, and he managed to pin down that it was tight junction proteins as marked by Z01 that was, or sorry, Z01 was really the key here for MLKL, because there's complete overlap with the phosphorylated MLKL signal. So this tells us that MLKL and ZO1 are likely engaged in trafficking via the same pathway. And it was also interesting to know what the effect of the neighboring cells were. So HD29s are adherent cells, and we saw an X and V positivity coming up, and then it seems to conduct itself to the neighboring cells. So perhaps via these junctions that are formed between cells, there's some communication at the level of lipid reorganization on the plasma membrane, which is conveyed to the cell. And so this is something that Andre painstakingly enumerated over the course of what may well have been a month. And he found that the cells adjacent to each other die faster. And so the important thing here is that if you label some cells that are without MLKL and some that are wild type, you need to have MLKL for the cells to die. So they have to be prone to necroptosis for this effect to occur. So I should have pointed out, sorry, that cytox is our marker for cell death, and this occurs after this membrane lipid reorganization that we've read out with an Nexin-5 binding. Okay, so with this, we kind of had a picture of what was going on, but it was incomplete. And one of the things that we've been doing was to generate some monobodies, so to try and dissect the pathway a bit further at the molecular level. And so we started a collaboration a number of years ago, and it was actually at a FASIV meeting that Natalia co-organized, and I got talking with Shohei Kawiti, and eventually managed to convince him to work with us on this. And I have to credit Jim Wells for putting in a good word for us as well. But this has been incredibly powerful. I think we've got some really cool tools now because of this collaboration. And I'm just going to change back. So the ones we have a library of them that I'll tell you about in a moment. But the key things here are that we found some inhibitory ones that bind to the N-terminal domain of MLKL. And we found some others that bind and that serve as probes for MLKL within the cells. So for those of you who don't know what monobodies are, they are kind of the same principle as antibodies, but they're based on a fibronectin 3 scaffold, and the library that Shohei and Akiko Kawiti developed years ago really introduces variable loops in different positions. They have 2 libraries that they use. The cool thing about these proteins is that you can express them in the intracellular environment, so they're not secreted. So you can use them to dissect signaling inside cells. And so what we did was we made some biotinylated proteins. So either the N-terminal domain, which as I mentioned is the executioner of MLKL, the regulatory pseudokinase domain, or 2 forms of the full length. So one was either a stable monomer and the other was a tetramer. And we were able to generate, I should say, Akiko and Eric and Shohei were able to generate a library of 42 monobodies that we subsequently went on to characterize. The first study that we did was with these N-terminal ones. So they bind with nanomolar affinity to the 4-helix bundle domain. And for all these studies, we used a C-terminal binding protein, monobody 32 as a negative control. And so this is just shown here. So what we're looking at here is we're either inducing expression with DOX. I think I've got the bars around the wrong way, sorry about that. So in black, it's plus DOX. And we're either expressing monobody 32 or not, and these are untreated cells, cells treated with an apoptotic stimulus, TNF and mimetic, or with a necroptotic stimulus. And you can see that monobody 32 has no bearing on whether the cells die or not. But in contrast, these N-terminal targeting ones, I'm showing you monobody 33 data here, but the same applies to monobody 37. In 2 different human cell lines, we can see no effect on apoptosis, but a very complete inhibition of necroptosis, which is very cool. And this is also species specific, which is what we'd expect based on the sequence similarity I told you about before. So the divergence between mouse and human, so this is in mouse fibroblasts, and we see no bearing on death in these cells. So we're also able to look at this from using a similar sort of assay that I mentioned before for imaging, where we can see that in wild type cells treated with the necroptotic stimulus, we see these hotspots of phospho-MLKL in the junctions between cells. So this is blocked in the case of where we have monobody 37 expressed. And of course, this is just a control where there's no MLKL. So this tells us something's happening to block, the monobody is blocking MLKL progress to the plasma membrane, or at least its assembly into higher order species on the membrane. And so this is something we picked apart a bit further. So using more conventional biochemistry approaches. So one thing we did was to express these proteins. So they're as GFP flag tagged proteins with a flag tag on them for detection. And you can see that when we express them, shown with DOX here, you can see they've come up nicely. And then we treat with a necroptotic stimulus, we can still see phosphorylated MLKL. So it's not impacting necroptotic signaling at this level, not at the engagement with RIP3 or the phosphorylation of MLKL level. The monobodies are still able to IP MLKL, and this means that it can you can still see phosphorylated MLKL bound by the monobodies. But then I guess the real answer came from a Blue Native page, which is a common assay we use for trying to study MLKL compartmentalization, because we can look at cytoplasmic versus membrane fractions, and we can look at whether MLKL forms into a high molecular weight species by a native page. And so normally MLKL under resting conditions is a lower molecular weight band. And here we don't have any necroptotic stimulus, so nothing happens. But when we do add a necroptotic stimulus, we see it changes from the cytoplasmic fraction to principally the membrane fraction and forms this high molecular weight species. And so this is just before we add, this is with monobody, but before we add stimulus. And when we add monobody and our necroptotic stimulus, we can see that it forms a high molecular weight species, but it's not membrane associated. So this tells us that we've trapped, with the monobody, we've trapped the protein in the cytoplasm. So this is something that we looked at structurally. So with Rich and Emma, so we were able to map the binding site onto the this alpha 4 helix of the 4 helix bundle domain. And so marked here in green on the right, where the known sites of lipid binding residues. And you can see that's on the opposite face to where alpha 4 binds. So this is still a mystery. We're not blocking the lipid binding residues, we're blocking a different process. And so we sort of tried to pick this apart with mutational analysis, and we could identify 3 key residues that were essential for this interaction. So our conclusion from this is that what we're doing by mutating this site is blocking interaction with an important chaperone for moving MLKL from the cytoplasm to the membrane. And so that's something that we're still actively trying to pin down. We don't know the identity of the chaperone, but we know it works via the Golgi actin microtubule pathway based on the imaging I showed earlier. But we're also interested in C-terminal domain. So we thought that the pseudokinase domain and monobodies directed towards them. So initially we hypothesized that maybe we could block necroptotic signaling by somehow blocking this interaction of MLKL with RIP3, but this proved to be disappointingly not so. But it turns out that we can use these as important probes for dissecting what's happening inside the cell nonetheless. And so 2 that we've been working on, I've already told you about monobody 32, that was in the last study as well, but the other one that we've been working on is monobody 27, and these bind with nanomolar affinity to the pseudokinase domain. And so Sarah Garnish has led this work, so she made this really cool observation, which was that, so while monobody 32 binds to MLKL rain or shine, so phosphorylated or otherwise, post-stimulus it's phosphorylated, monobody 27 doesn't normally bind to MLKL. It's only after it's phosphorylated post-stimulus that you see this interaction. So this is really cool because it told us that we've got a cellular probe for activated MLKL for the first time, which is really cool. And she'd also managed to show that in RIP3 knockout cells generated by Annette in the lab that you could see MLKL binding when you take away RIP3. So it seems that MLKL is bound to RIP3 under basal conditions, which prevents access to monobody 27. And this was a really cool observation, and this happened, almost immediately before we went into our first lockdown. So it was sort of a tantalizing observation, but that didn't slow down Meng, another PhD student in the lab, who generated a number of complexes of monobodies with the human MLKL pseudokinase domain. And just before our institute closed to working from home, he submitted to our crystallization facility some complexes. And within a few days, we had these beautiful crystals, but we were just sitting at home with no synchrotron access and no way to recover the plate. So we had to wait patiently for another more than a month in order to shoot them. But it was worth the wait. So what he found was monobody 27, the one that only binds to activated MLKL, binds in this sort of front-on way to around the activation loop. And we can see that it binds in the conventional activated protein kinase-like conformation, where we've got this sort of closed conformation in the invariant lysine and alpha-C glutamate in this interaction, and R spine's intact. So typical of an activated-like conformation. And also what we saw previously with an earlier structure that we solved of human MLKL in an apo form. So this told us where the monobody was binding, and it bound only in this activated form, as I mentioned. And also Shohei was particularly excited because this was the first example that he's aware of a monobody binding to an active site, or in our case, a pseudo-active site. And so, then we started to rationalize why we'd only see monobody 27 binding after MLKL is activated. And so the best lead we had was really to superimpose with the only known MLKL RIP3 complex structure, one from Yigong Shi from a number of years ago. And so this is our best guess as to how the human complex looks, because we don't have an experimental human complex structure yet. And you can see that they're adjacent and in part overlapping. So this sort of tells us that maybe there's some sort of competition between the two for binding. And this is something that Mung's been able to go on to show biochemically subsequently, which is lucky because a reviewer or two have asked for that data. So this gives us a really good lead on what the activated form we think of MLKL looks like with this active kinase-like conformation and an unstructured activation loop. And it also gives us the way we can rationalize this is that we need release of MLKL from the necrosome before it can be detected by Monobody 27. So we think this is a new signaling checkpoint, or at least formal evidence for something we've speculated on for a very long time. So the other one that we've been interested in is Monobody 32. And as you will have seen, we've used this as a control along the way. And not within our wildest dreams did we expect the structure with MLKL would be very interesting, but it turned out to be incredibly interesting. So it's another one that Mung was able to solve. And what we saw was the activation loop forms this helix shown here in green. And also the full activation loop was able to be observed in this structure. And for the first time for an MLKL pseudokinase domain. And this isn't entirely dependent on the monobody because this is binding at the spine, not near the pseudoactive site per se. So from this, what we've inferred is that this is likely to be the inactive basal form of MLKL. And I should point out that Monobody 32 binds MLKL regardless of conformation in cells. So we were probably fortunate to capture this conformation in the crystal. But just to compare with some of the other work that we've done, so we've got our Monobody 27, the form that we think is activated, and then we've got this basal, what we presume to be an inactive form that was captured by the MB32 complex. And we can compare this to the activation loops of other orthologs of MLKL. So really the key point here is that mouse MLKL has this activation loop helix as well. And this is something that we observed a number of years ago. And at that time, we proposed that perhaps this represents one phase of the conformations, and it can toggle to this phase, which we saw with the human structure. And so now we at last have some formal evidence that these two conformations can occur. And Catherine Davies in the lab recently published a paper looking at rat and horse MLKL, and rat kind of looks like human, but that even though it's like 90% identical to mouse. But the horse one's cool because the activation loop runs into the ATP binding site, which we think is an unprecedented binding mode. So I think what this tells us is that activation loops can be highly variable between the different orthologs, and that it probably is one of the key triggers in determining activation, principally because it governs whether there's this release from RIP3 or not. And this is something that we looked at computationally with the help of Cannon and Weilin Leung in his lab. And so our first hypothesis was really that this helix that was formed in the Monobody 32 structure would be perturbed by phosphorylation. So when RIP3 phosphorylates the activation loop, we'd expect to see this helix disintegrate. And this isn't what was supported by his MD simulations. Instead, it looks like the phosphorylated form's quite stable. So even if you were to phosphorylate this activation loop helix, it would still persist. So we looked at something else, which was that perhaps the phosphorylation event reduces an energy barrier to this proposed conformational interconversion. And that's precisely what we see. So if we look at the two minima from the Monobody 32 structure and the APO structure, and then we're looking at this trajectory, looking at the distance between the invariant lysine and the alpha C glutamate. So we're looking at the conversion to a conventional active-like kinase conformation, and you can see that the barrier is lowered in the phosphorylation, in the phosphorylated MLKL simulation. So this tells us that the interconversion is more readily achieved upon MLKL phosphorylation. But this really supports the idea that that we've previously explored with phosphomimetic mutants, that MLKL a phosphomimetic mutant of human MLKL doesn't bind to RIP3 in SPR. So this sort of tells us that this is likely, phosphorylation is likely to be a release switch, and this seems to be accompanied by this structural interconversion. So just to summarize, so what I've shown you today is a bunch of new tools that we've generated that we're very excited about. And this has allowed us to sort of pick apart some more detailed aspects of the pathway. And some of these things have been used on for a number of years, and we've certainly written about them in reviews without formal evidence. So one is RIP3 disengaging from, MLKL disengaging from RIP3, MLKL oligomers undergoing trafficking, and then the necessity of this accumulation into hotspots for cell death to occur. So the other thing that I think is very cool is that we've been able to obtain formal evidence for human MLKL existing in two conformational states. So this adds to the idea that it can function as a molecular switch, which can be toggled by RIP3 phosphorylation. But we don't know everything, of course. So we still don't really know what the Q is, which governs whether how MLKL is recruited to RIP3. We don't really know what the chaperone here is that's mediating via alpha-4 in the alpha-4 helical bundle that's mediating this trafficking to the membrane. And it's almost assured that there are going to be other proteins involved in the pathway, and we're actively trying to find their identities. So lastly, a bunch of people to thank. So the Monobody 32 and 27 story is the work of two people who have just completed their first year of their PhDs, have done amazing work. And even with the challenges of COVID, we've managed to get somewhere. Andre, who has led the imaging aspects, showed you Joe's story that she did with Maria, looking at the PLT15 mouse. She did some Catherine's work. We've had great technical support from Sam and Cherie over the years. And Annette's PhD student has done great work. Chris, a postdoc, and other PhD students and technical staff. Alexandra works on a different project. Lung Yu, another project as well, another pseudokinase project. Daniel, who's completing his PhD, and Wayne, who's a summer student who's contributed to a lot of this work very efficiently. And Emma led a lot of the monobody work, and she's now left to a different role. And we've had a number of people through the lab who've contributed. Some great collaborations at WeHi, without which none of this would be possible. The core facilities as well, so imaging, the antibody facility is essential to the work we do. Mass spec, the screening lab have contributed. And of course, the monobody story couldn't have happened without the help of Shohei, Kiko, and Eric. Modeling collaborators, Cannon's lab, including Wayland. And we've had some help with phylogeny analysis with Wayne and James, and also thanks to our funding bodies. And thank you for your attention.
Thank you, James. This was a really wonderful story. So nice to see how the pathway gets more and more delineated and specific. So I encourage everyone to type their questions into the, you can raise your hands too and ask your questions. That's fine too. And I hear someone has, Adil, would you like to ask your question now?
Hi, thanks. That was a great talk. This is a lot of work. I had a question regarding your microscopy. So it might just be the images with the representative images, but it seems that you see a lot of the hotspots for MLKL, and those hotspots seem to be for phosphorylated MLKL, sorry, but they seem to be in the area where there's two cells contacting each other. So I was wondering if you thought cell-cell contacts were required for kind of that threshold of phospho MLKL required to kill. And if you had one cell that was like wild type for MLKL and one that was negative, do you think that a cell that acquires that threshold of phospho MLKL at the membrane could kill that MLKL null cell? Thank you.
Yeah, okay. So yeah, good question. So the first one is about the junctions. Yeah, we think that it's not uniform. I mean, some cell types don't form junctions and they can still undergo necroptosis. So it's just really an indication that in that cell type and some others that we've looked at, as Andres reported, have this common trafficking pathway with the tight junction proteins. So we think that's really the basis for it. And he shows in his paper that if you weaken tight junctions with pharmacological agents, then you can lower the threshold to necroptosis. So clearly it's important in some contexts, but not all. The other question I've forgotten, whether you have a knockout, MLKL knockout cell and a wild type cell. He's done that experiment, so he labeled them differently, and he saw that you need to have an MLKL replete cell as the adjacent partner in order to see death to be conveyed to it by that membrane remodeling. So you do need the cell to be prone to undergoing necroptosis for that to be conveyed.
Following up on this, James, I was wondering about this is really quite remarkable that you have this accumulation of MLKL at the cell junctions. And is there is there a transmission of some signaling mediator across the cells? Do you think something gets transported?
Yeah, that's a really good and important question that I don't have an answer to, but it'd be really it is something that we're interested in knowing, whether there is some sort of conductance between adjacent cells. So yeah, we don't know the answer, but it's a very distinct possibility, because it seems that one cell can condition another to undergo necroptosis. But more importantly, I think if we've got this membrane remodeling occurring, which is read out by phosphatidylserine exposure with the annexin-5 staining, then we don't really know what the purpose of that is in the context of necroptosis. So others, including us, have observed that, including Doug Green's lab and others, that Modi Gerlich, that if you stimulate cells to undergo necroptosis, you see this annexin-5 binding occurring, staining occurring well before cell death, but we don't really know what the function is. So there are ideas that maybe MLKL has a separate function as a scramblase, or maybe it's conditioning the membrane somehow to be permeabilized. These are all things that we don't really have a molecular knowledge of, though.
Yeah, it's really fascinating. I see Ed has a question. Ed, do you want to ask it? Is it possible to get Ed to ask his question?
Yeah, can you hear me now?
Yes.
Yes.
Nice to hear from you, Ed.
Yeah, beautiful work. The question is about whether the MLKL clusters are forming in the cytoplasm.
Yeah, so that certainly happens. So we do, it seems to be a more modestly sized, as far as we can detect. Of course, we're only, at the mercy of the reagents we have, but it seems to be more modestly sized oligomer. And so we think they're happening at necrosomes. So now we have some more images coming through with, now that we've got more reagents to look at this. So we're able to stain for RIP1, RIP3 and MLKL together. And so we can show that these accumulation events start at least at the necrosome, which is a cytoplasmic compartment.
More technical question about the monobodies. How were you able to generate the ones that are specific for the phospho form?
Dumb luck. That was what's so great about it. So Sarah, being as diligent as she is, just tried all of them, and we thought there's got to be some gold in these here hills, and so she went through the motions of trying all of them, and we're just really lucky that one of them ended up being a conformation specific probe. So I guess luck and hard work on her part.
That's awesome. I see Jeremy Thorner has his hand up. Can we get Jeremy to ask his question?
Can you hear me?
Yes.
Yes.
Okay, great.
Hi, Jeremy.
Greetings across the bay from UC Berkeley. Quick question just about after the need for ATP for the RIPK3 phosphorylation of MLKL, do you need ATP, sort of an analogy to what Xiaodong Wang found for DATP being needed for apoptosome oligomer formation? Do you need ATP binding to the pseudokinase domain at that point to get oligomerization, or is it irrelevant?
It's a good question. One that we've looked at both biochemically or biophysically and in cells using mutant protein. So we know that ATP binding certainly promotes the formation of like modest oligomers, tetramers. But in cells, when we put the mutant in, it sort of retarded cell death, but it didn't necessarily completely knock it out. So we don't, even though MLKL can bind ATP, we don't think it's essential to its function.
Yeah.
We have one more question from Erin. Maybe I'll just read it. What are your thoughts about why some cells have the hotspots and others don't, but still undergo necroptosis?
Oh, so the hotspots do occur. They just don't occur at cellular junctions, and they're obviously more modestly sized. So we still do see an assembly of phospho-MLKL on the plasma membrane. It's just not as localized as to the cellular junction. So maybe it tells us that depending on the composition of the plasma membrane, they may have different susceptibilities to permeabilization. But it's obviously only something we can speculate on because we don't really know precisely how MLKL permeabilizes membranes. We just feel that maybe it's to do with disruption rather than a formal pore. But yeah, that's a good question, and yeah, one that we don't really have an answer to, like so many things.
One more clarification question. So you think that phosphorylation of MLKL, do you think it's an important step in disengaging from the rib case, but also plays a role in trafficking and oligomerization?
Certainly the first part. I mean, with the viral MLKL, which is a competitor for cellular MLKL, it binds to RIP3 and sticks there, and it doesn't have an activation loop per se, so it can't be phosphorylated, so it can't disengage. So it basically sticks and prevents cellular MLKL from being bound and activated. So that's one sort of, I guess, anecdotal piece of evidence for why we think the role is really to do with disengagement. Whether it is involved in promoting oligomerization is something that we're actively trying to understand. So we are trying to generate structures of phosphorylated MLKL, and I believe Mung has one of the pseudokinase domain now, which is phosphorylated, and it looks like that active conformation that I proposed, which is the closed conformation. So I guess, we'll learn. We need more. So part of the problem is that generating reagents to human RIP3 has been really problematic because the protein is incredibly unstable in isolation. So we've got a few ideas on how we might be able to generate Fabs now, but hopefully with that sort of those sorts of reagents available and perhaps some better inhibitors that a number of people are reporting them now, we'll be able to get somewhere with experimental structures. Great.
I see no more questions, so I think we're gonna wrap it here. And thank you, James, so much again. That was really spectacular. Really great to see what has happened during the pandemic. Amazing.
Yeah, I'm glad it went okay because I haven't spoken for about a year, so feeling a bit rusty. But anyway, yeah. Thank you. Thanks. It's been
it was a lot of great data. Thank you. Thank you. And we'll be in touch.
Great.
Hopefully,
we'll keep in touch.
See you soon.
Yeah, hopefully in person.
Yeah.
Right.
Yeah.
All right.
Take care.
Thanks. And good luck to everyone with your election.
That's right. Bye, everyone. Thanks for joining.
Thank you. Bye.
Bye.